Thank you.
Right? Okay.
Good morning and welcome everyone for today's session of Indian webinar series Introduction to Computational Drug Design, co-organized by Schrodinger and PCI. I'm Shelvia and I'll be your host today. I would like to take a moment to thank our collaborators for your support for this event. Today we have another demo session and induced with docking and covalent docking. If you have any questions during the course of this presentation, you can drop your queries in the chat section of YouTube link, and we will have a Q&A session at the end of the session to answer your queries. Without further ado, I would like to invite our speaker, Dr. Vinod.
Yeah, thank you, Shelvia. So let me just try to share my screen and hope you can see my screen now.
Yes.
Place it in presentation mode and get started. Is it fine?
Yes, it's fine.
Okay, thank you. So very good morning to everyone. Thanks for joining for today's demo. And I'll be talking and I'll be giving a demo on induced fit docking and the covalent docking. I have two case studies on induced fit docking, and I'll be covering one on covalent docking. So how it is and what are the difficulties we face and when we need to use it. And for the theory and presentation, I would request the viewers to check for this YouTube video with this topic, virtual screening to prioritizing the molecules and need for flexible docking and covalent docking. The second half has my presentation where I have covered the complete theory on covalent docking and induced fit docking. So please, I request you to go through this. And what would be my consideration is just to get started with, we have induced fit docking, right? So I think I clearly mentioned it in my previous session that induced fit docking is nothing but inducing flexibility in the binding site. And why you need to do this? I clearly told you the various scenarios, like when especially you have an active molecule or a molecule that is well known to be active and the crystallographic structure is not able to accommodate, and you have various flexibility criteria in your receptor, or you want to induce different loop refinements, then IFD is a good choice because it simultaneously performs glide molecular docking and prime loop refinement in a stepwise workflow. So as this all sessions are specific to demo, so I will talk more on demo. The first case study I would be exploring is induced fit scenario of a DFG loop in MAP kinase. So MAP kinase has this tricky DFG in and out motif, and there are, like, this is just an example. So different conformation, active form, inactive form. Certain selective inhibitors are better than the other. So let me just show you my Mastro. Hope you can see my Mastro, and then we'll continue. So here you have the Mastro, which I have launched. And the first thing that you need to do anytime, whenever you work, you have to change your directory. Change directory, and I have this folder specifically dedicated for the demo. And now I'll just explain you the problem. The problem is right now I have is, let me just open that project, and that would be easy. That is file. I have saved everything in a project. So here is a project. And this is a ligand that is 1kV1. You can see this is the molecule that you have in the workspace. Let me show you the crystallographic structure of this. This is 1kV1 molecule. Okay, it's in raw form, unprepared form. What are the problems you have? One among the problem that we face right now, it is like obviously you can perform protein preparation wizard to fix all the problems. But let me just show you like what is the problem currently, the major problem, which is the brakes. If you notice here, there is options to activate the chain brakes, and you can see that chain brake is in the main region of DFG motif. It has a bound ligand, and this DFG motif is very, very critical. So you can see this is the location, and here is a gap. Like you can see the residue is here it is 170 and here it is 184. So there are missing amino acids here. And what you need to do is you need to perform protein preparation completely to remove that problem, and you can get the structure like this. But my understanding is, okay, let me take this ligand and try to perform induced fit docking with another MAP kinase bound inhibitor, that is 1A9U. This is a molecule that is already bound, and I have already prepared. This does not have any gap. If you notice, this does not have any gap. You can see clearly this is the molecule, this is the molecule, and it doesn't shows any gap in between. You can't see any breaks. If you see a break, you can see red and blue color there. So you need to prepare the structure fully. Here is the option for seeing the structure which is completely prepared. So how do you know whether your structure is prepared or not? Usually in Schrodinger, whichever has the energy parameters, like if you see in project table, there are energy parameters that shows you whether it is prepared or not. In property, you can check. I can show you if you want. So we have impact. So you can see the energy parameters. So that means this structure is prepared. Now, as I said, so you don't take a lot of molecules. So it's very clear that I shall be taking 1 kV1, and my objective is to dock this against 1A9U, okay, which is also prepared. Now, what is the benefit of doing this? Okay, the main ideology is to explore this DFG motif, which I was referring to. This motif has DFG in and out conformation. So I will show you that, but before that, let me show you how to set up the calculations. So you need to select the ligand of your choice and include the protein of interest. Okay, so what you will do, you will go to task, browse, and we have induced fit docking. So there's a much advanced method as IFD-MD, as I told you in the previous presentation, but we will purely focus on induced fit docking. The first thing that I need to do is I need to mention the ligands to be docked in the project table selected entry. Here I have mentioned. So you can now notice here it is selected. The protein only is included. In the bottom also you can see the status, one selected and one included. There are two modes of IFD. One is standard protocol and other is extended sampling protocol. Extended sampling has the tendency to give close to 80 poses, and the standard has tendency to give 20 poses. If you notice, when I go for extended, the steps are predefined. We don't have to customize everything. But let me show you the standard protocol. I clearly told you that it's a combination of two products, and that is between glide and prime. Okay, so here I have to define the receptor. Let me try to define the receptor. Whenever you have structure in the workspace, just double click on preset so that you can see the ligand clearly. The ligand is visible now. And once the ligand is visible, now I can mention the grid centroid. So that you can do by clicking here. Now the grid is defined. Now what it will consider is all the loops that lies within the grid. It is going to perform loop sampling and simultaneously docking of that. In case you don't have the centroid of the workspace ligand, there are options for specifying the residues. So how you can identify the specific residues? You can go for sitemap. Sitemap will evaluate all the possible binding site and then it will give you the binding site residues. Don't give individual biased residues. Give residues of the complete pocket. Then the grid centroid would be much better than when you give individual residues of your interest. Because molecule need to bind to the receptor pocket and interact with your residue. It doesn't mean that you have to force it to interact with your residue. The problem comes when you perform MD. That's the reason many of the molecules end up not being stable in the binding pocket. If your residue, specified residue, is not properly the centroid of interest. Then you can mention whatever constraints you would like to have. We can mention the conformation sampling of your ligands. And here you can perform specific trimming by using side chains or constraint refinement. And here is the options for specifying the loop of interest for loop refinements also. So you can specify which loop so you want to refine. Otherwise, by default, it will sample all the loops in the binding grid. So that is the reason it takes a long time, and which residue you don't want to move also can be specified. The glide redocking, currently you can see there is SP option, extra position option, and simultaneously you have options for writing the XP descriptor information also. Okay, so this is all set. I gave you many things, but let me just keep it as simple as possible. I will reset and then redo this setup. So as I said, I need the protein of interest, which is purely prepared already. And the second is the ligand of interest. So ligand to be docked, I will mention here. I will extend it and I will pick the ligand. That's how easy it is. So when you go for your job run, you can distribute it into multiple processors. Like I can give here 8 and 8 here. So when I hit run, then the calculation will be done and it is going to give me the output. So that's how easy it is to launch a induced fit docking. Now let us try to analyze the results. I have generated more conformers so that you get better view and understanding on this receptor. Now if you notice, this is the output. You get different complexes which is being sampled, right? There are so many posers. Now I will try to play it as a movie so that I get a better view. Let me try to do that. So here is an option for single direction. And then when I play this, please notice that there is some movement in the binding site. Because the algorithm is bound to do this, right? I clearly told you it will perform loop sampling in the binding site, and you will be able to visualize the flexibility of the ligand molecule inside the receptor. So this is much accurate than our molecular docking approach, but it is computationally intensive and costly. So that is the reason we don't use IFD for virtual screening. Okay, so how do you analyze it? The first thing that is very famous with industry is the IFD score. IFD score, you can see it in project table also, or else you can see it in the entry list also by specifying here the custom properties to show. Now the value should be as negative as possible. This is going to be the sum of prime energy and the docking score and all that. But you should not just consider the top pose. It's not about that. It's to consider the top 10 or 20 poses simultaneously. Now, how do I analyze? The analysis is like to see the consistency of the pose and notice what are the changes that happens. Now, when you see here, here is your DFG motif. Like how I will show you. Here is your DFG motif. Like if I just pick any one residue, you could see in the sequence viewer that is selected. See here, it is selected. Now to select the complete DFG motif and visualize them, what you can do, you can come to define and then just say selection. Because I have included the top 10 poses. And once I do that, when I say OK, you could, let me just see, selection and residue number and OK. So now you could see the complete batch of residues of DFG motif is visible. Can you see here? Okay, just to visualize only the ribbon view, I will hide everything else. Okay, so I have hidden everything else. So clearly you can see the in and out conformation of your protein. So how this molecule is available in the DFG in and out motif is something of my interest, because now I can see what kind of consistent interaction it holds. So this 10 poses, when I add the ligand molecule or the surrounding amino acids, just double click on here and you can see the complete surrounding amino acids. And there is huge flexibility in the binding pocket. So whenever you analyze IFD poses, it's not just one pose. It's not just top pose. It is about consistency. Like out of 10 poses, what are the frequent interaction that has occurred? How you can visualize that? It looks a lot of interactions, right? So you will notice now where are the frequent interactions. You can see the web of yellow dotted lines and which residue is frequently interacting in which location. If it is little difficult, I'm just removing the non-polar hydrogens. So here you see. So that is the reason you can see the loop movements and also the conformations of the ligand. I know it is little difficult to analyze in this way. So that is the reason we go for SIFT. That is fingerprint analysis of the complexes. You have to go here and in this you need to mention, select the top poses and select this option, receptor ligand complexes. In docking already you have seen this panel, but there is an option separately, and if you hit generate fingerprints, then you can see the matrix of interactions. So here you will be able to visualize what are the consistent interactions that are available. At one go, we will be able to conclude like, okay, this molecule is making these consistent interactions. The other thing that many of the research scholars do is take the top pose and go for molecular dynamics. And even you can calculate via script the binding energy of all these poses that are generated from induced fit docking. You can also perform enrichment like ensemble docking, because these are the various conformers of the receptor, right? There is a concept of ensemble docking. And ensemble docking is done on various conformers of a single protein. So that also you can perform. So that's how you induce the flexibility in the binding pocket. And if I have to show you what is the difference between the site, like in case, okay, we know you have done docking. Now I can see here it is 1A9U. And this is 1KV1. So what is the difference it has caused? What is the change in the binding site? So that can be done by using sitemap. Sitemap is a nice tool to evaluate either a single binding site, or you can identify the various binding sites in the complete pocket. Now, what you will do is you will try to pick the single binding site. Just go and pick the ligand molecule and it is going to evaluate this binding site for you. And now I have done for 1KV1 the binding site analysis. This is the outcome I have got. And after IFD, so this is IFD results, and this is before IFD. If you see, these are the two results you can notice here. So this is before IFD, quite exposed, and you see here this is after IFD. And if I try to show you in a tile view, this is how it looks. You can see clearly, and it has a break. If you remember, it had a break, and after IFD, you can see your pocket is well conserved, and your virtual screening will have better enrichment. So that's how IFD need to be taken care. Now I have one more example. I will just show you that too. The second example, what I have is CDK2 receptor induced fit effects. So this is a straightforward case. I'll open that project, that is CDK2 IFD, and you will notice like this is the input receptor CDK2. Okay, so here is a bound ligand already. You can see the ligand, how it is bound. And I will take the ligand from CDK to 1KE8. So this is much, you can see much bigger molecule, right? And it is different than this molecule. Different heterocyclic nucleus induces different conformation inside the binding pocket. And our goal is to identify bioactive conformation of the protein ligand complex. So you should not get confused with global minima conformations or local minima conformations. I'm talking about that complex that is in bioactive form. So that is going to help us in finding out new molecules, new and better and high affinity molecules. So here is your target receptor with 1KE8. And once I do what I showed you, right, in the induced fit docking, browse, and here is your induced fit docking. I need to do the same way what I did, so I'm not going to repeat that. And the output that what you're going to get looks something like this. So you can see the induced fit scenario of this molecule and note down what are the consistent interactions of this molecule against different poses of IFD. Again, I said, in case if you're planning to do molecular dynamics after IFD, okay, so this flexibility to attain or loop movements to attain in MD, you need to run in a very long stimulation. So in prime, the benefit will be the loop conformation is specifically done. So the stability that it attains in MD would be much, much easier. And let me try to show the similar analysis of shift of how it looks. And here is your consistent interactions. So if you see whichever the consistent interactions are there, you need to note it down, and you need to mention that like these are the frequent interactions that need to be taken care. Whenever I keep my cursor, you can see in the here the interaction residues are mentioned. So this is any contacts you can specifically get interested with H1 donor or acceptor and all that, and decide, okay, these are the critical interactions that I need to focus. So this is the second example. So that's how you need to use induced fit docking. And I will move forward now and let me go to the next topic, covalent docking. Okay, so drugs that forms a covalent attachment to the target, again, it uses both the products, that is enzyme plus inhibitor, initially forms a non-covalent complex. And if the receptor has the tendency to make that interaction, then obviously it will form a covalent bond. So not all receptors tend to form this kind of covalent bond. Now, if you notice, the shallow binding site will be used, and the receptors should have the tendency to form this. And usually, tyrosine, threonine, and cysteine are the ones that forms the covalent bond. Now, these are the reactive warheads, some of the reactive warheads that is going to be used. And there are various reactions, well-known reactions. And I'll show you the covalent interface. And let me explain, this is how the covalent bond forms. I'll show you the demo now. So I'm just closing this project, and I'll open File, Open Project. So here is a project which I have saved. So the receptor is 4XAM. If you notice, it has multiple chains. Now, whenever you have multimer, it is okay to retain just a single chain. So I have retained it, and I have performed stepwise protein preparation, and I got this 4XAM. And if you do preset, you can clearly see where the covalent bond is formed. This is a triazine nitrile warhead, okay, and it forms a covalent bond with cysteine 25. I think it's visible. So that is and I have set off close to 8 molecules where you will notice all of them have the same reactive warhead. The library, like it's a small thing that you need to focus is the library of molecule that you use for covalent docking should have same warhead, reactive warheads. So you can see here triazine nitrile is a reactive warhead I have, and it forms the covalent bond by nitrile addition. Okay, it's a custom reaction. I will show you that. And so here is a receptor before preparation, means I have removed it. The ligand is removed, and you can see the cysteine alone. And this is how it looks. In case you want to do molecular docking on a covalently bond molecule, it is important that you delete this bond. Okay, you have to delete this bond, and you need to be careful in like how you do this deletion. Okay, so you have to select this, and come here, then there is an option of deleting the bond. And then you can perform your normal docking, because again, this is a frequent question that has been asked from me. So that is the reason I had to show this. Okay, so this target is cathepsin L inhibitors. Okay, and how do you enumerate a library of your molecules? Like for example, now I have this molecule. I need to enumerate a library of molecules from this receptor. And how I can do that? So for that, because you can't take online databases, because your reactive warhead is very specific, right? This reactive warhead need to be. And if I do a random enumeration or R group enumeration, like many of the software has, like in this case, now I've taken two groups. Just imagine I have taken these two groups. I can go for build, add fragments, create enumerated entries. The problem is the number of synthesizable molecule will drastically go down. The practical molecules that can be synthesized will go down. So that is the reason I don't suggest automated enumerations. Rather than that, you need to go for reaction-based enumeration. So what is the option for reaction-based enumeration? You have pathfinder and reaction-based enumeration. So this will take the molecule and just you have to hit load. See, this is the molecule and you want to retain this, right? Triazine nitrile group, because that is your warhead and you have proper literature. So what you will do, we will just hit generate pathway. It is going to give you different mechanism by which it will generate the reactions. In case if you are aware of your reactions, then you can use it and you can sketch it. You can sketch the pathway and then use it. So the reaction library, what are the reactions library that are available? It's also mentioned here. So for covalent inhibitor research, this is very, very tricky. Now, when I try to build this, I will end up in getting lot of molecules. Can you see here? There are close to 999 molecules which I have generated by using this reaction-based enumeration. And all of them consist of this reactive warhead for my ligand molecule. Now, I told you, I think there are two sets of problem. When I have small set of molecules and when I have large number of molecules. Now, what I'm going to do is I'm going to select this 8 molecules and take my fully prepared 4AXM. It's very easy to define and perform covalent docking, but in many of the other software, it's very, very difficult. So what we need to do is we need to search for CovDoc. So we have covalent docking. The covalent docking tool name is CovDoc. And the ligands need to be selected like what you did in the induced fit docking. And once you have to pick there are two things that you need to pick. First is the reactive residue. I think I told you this is 1625. Here it is. So here I have picked the molecule and then you have to pick the centroid of the ligand for the grid. Now, if you do this, this is the first setup with respect to the reaction. Then the second setup is to choose the reaction type. So by default, Schrodinger has many common reactions. You can see here, but this is a specific reaction and it's very easy to customize your specific covalent interactions. I have already customized this nitrile addition, so I will take that file. So it has taken it and evaluated and found the sites for the same. It is pretty smart. Like if I choose some other thing, like it will identify whether any specific sites are available or not. You can see. So you don't have, it's not mandatory to have complete understanding on your reaction. Automatically your reaction library, if that has sites, then it can be taken. So I'm taking a custom file, I told you. So it is going to show me this reaction. Now, the next thing is I'm not going to use any core or constraint or torsional constraint. Next is docking. There are two modes. One is post prediction and next is virtual screening. So post prediction is good when you have handful of molecules. Like in this case, I have 8 molecules, but if you're planning to dock this 999 molecules, then you need to change it to virtual screening mode. So it will automatically analyze the complete molecules 999, and when you hit run, your covalent docking can be launched. So once that is done, you will see the result. So these are the, again, it like IFD, you get various outcomes. Like you can see the various poses that is covalently bound. You can see the complete poses. For a better view, I just set it up. You can see here is the view of the ligand molecules and how it is bound, what are the interaction it holds. And the molecules are usually by means of a score called as CDOC score. Okay, there is something called a CDOC affinity, and by that we decide whether the molecule is good or bad. Let me import it and show it to you. So there is a out file. This is how the files are generated. There is a out file that gets generated. And once you are done, I will show you in project table. So can you see here? CDOC affinity is visible. So you will get this score. The more negative will be the best for your design. So according to the current research, this molecule has a better tendency to form the covalent bond. So that's how you need to work on different covalent inhibitors. That was the demo on cathepsin L covalent inhibitors and advanced options, as I said, in covalent inhibition design. If you want to rank order, you can use FEP plus covalent inhibitors. So the concept is like it will take the ligand A in peptide and ligand B in peptide and then ligand A in complex, then ligand B in complex, and it will perform free energy perturbation. So those options are available, but it's again specifically for high computing resource, and the license is specifically available only for commercials. So that's all what I planned for today. I'm ready for any questions. Thank you.
Thank you for the presentation. We have received one question.
Yes.
As a reminder, if you have more questions, please add them to the YouTube link and we'll add them to our questions. So the question is, in what cases do we consider IFD as compared to molecular docking?
Yeah. So as I said, I think this was discussed in the previous session. Like when you have a homology modeled protein, then you need to perform IFD. When you have an active molecule and you don't have crystallographic or induced fit scenario, because every molecule will have an induced fit scenario or a crystal structure that does not have bound ligand. So you need to dock the highly active molecule and induce its pocket so that it opens up before the virtual screening. For example, I have seen many students working on protein that are not having any bound ligand. In that case, if literature reported not the crystallographically bound molecule, you can perform induced fit docking. And if you're planning to like explore the loop conformations in the binding site and see how much flexibility it holds, so that in that scenario also you can do induced fit docking. Means before MD also you can do induced fit docking. So, but otherwise many of them take it directly without analyzing. Okay, in MD alone, if you try to do, it takes long time for such movements to happen. Yeah.
Thank you for that answer. I don't have any more questions for you for now. So what I'll do is I'll just quickly move on to the announcements. If people have more questions, maybe we can come back to
Thank you, Shelby.
this one. Thank you so much.
So you need to share your screen, is it, Shelby?
Yeah, actually I received some more questions for you. So I'll just ask those first. Okay. Can covalent docking for non-enzyme glycation and cross-linking be performed?
Yes, the options are available, but I have not worked on those cases. But if you are interested, you can email us and we can get more details and I'll let you know.